On the beginning of the lecture, we look at the cell membrane, also called cytoplasmic membrane or plasma membrane. We also covered passive and active transport, and now it's time to look inside the cell. We find there jelly-like substance called cytosol with variety of little organs called organelles. Cytosol with organelles we refer as cytoplasm. Nucleus is not considered to be part of the cytoplasm as it has its own plasma called nucleoplasm. Nucleus is the most prominent organelle of the cell. It is most easily stained, so it was described early in the history of cell study. The nucleus is where DNA is, and DNA contains the organism genes, which control its heredity. People did not always know that, of course, but it was realized quite earlier that the nucleus was somehow important. It was found that if an amoeba is cut in half, the half with the nucleus will move around and survive, but the half without the nucleus will simply curl up into a ball and die. Nucleus controls all cell activities, including reproduction. However, the nucleus of an individual cell is almost as featureless in an electron microscope as it is under the light microscope. However, its membrane is interesting. We found that the nucleus is enclosed in a double membrane. Each of the two parts of the double membrane is regular lipid bilayer, so altogether there are 4 layers of lipid molecules and the usual associated proteins and carbohydrates. The inner and outer nuclear membrane pinch together in scattered places over the nuclear surface to form nuclear pores. Under the electron microscope, these pores look just like holes communicating between the nuclear plasma inside the nucleus and the cytoplasm outside the nucleus. We might have expected such pores since we know that very large molecules, the ribosomes and the messenger RNA molecules, must travel from the nucleus to the cytoplasm. We also know that all protein molecules that are found inside the nucleus, and there are many, are synthesized in the cytoplasm and must find their way in, presumably through the pores. It would make things a lot easier to understand if these pores were just holes, but they are not. It appears that in life they are filled with large globular proteins. They form nuclear pore complex known as nucleoporins. These proteins regulate molecular traffic through the envelope and act like a rivet to hold the 2 unit membranes together. Hundreds of molecules pass through the nucleoporins every minute. Coming into the nucleus are raw materials of DNA and RNA synthesis, enzymes that are made in the cytoplasm but function in the nucleus, and hormones that activate certain genes. Going the other way, RNA is made in the nucleus but leaves to perform its job in the cytoplasm. Immediately inside the nuclear envelope is a narrow but densely fibrous zone called nuclear lamina. This is composed of a web of protein filaments. It supports the nuclear envelope and pores and provides point of attachment and organization for the chromatin inside the nucleus and plays a role in regulation of cell life cycle. Abnormalities of its structure or function are associated with certain genetic diseases and premature cell death. As I mentioned earlier, the material inside the nucleus is called nucleoplasm. This includes chromatin, fine thread-like metacompose of DNA and protein, and 1 or more dark staining masses called nucleoli, where ribosomes are produced. DNA or deoxyribonucleic acid is the genetic material often described as a chemical blueprint of the organism. It determines everything from sex to body build and hair color in every human being or any other organism. The importance and function of DNA is discussed in lecture 13. What you perhaps notice on one of the previous diagrams is that outer nuclear membrane extends into the endoplasmic reticulum, often abbreviated as ER. Endoplasmic reticulum literally means little network within the cytoplasm. But before we talk about endoplasmic reticulum, let's take a peek at what's happening to chromatin before cell divides. You might have heard of chromosomes, but some might ask me, what is a chromatin? You see, chromosomes are formed during cell division. Many cells in living organisms, including humans, has to replicate. They do it by dividing generally in two cells. One parent cell divides into two daughter cells. Chromatin is a thread-like matter. The goal of the cell division is to eventually divide genetic material between two new daughter cells. But how do you divide something that is thread-like and easily tangles? Is there a solution? Yes, there is. During cell division, chromatin condenses and turns into short structures called chromosomes. Do you see it in the diagram in front of you? Chromosomes in the nucleoplasm. Why chromatin has to turn into chromosomes? Because short structures are easier to manipulate during cell division. Cell division is covered in the following lecture, number 11. Here's the structure of a chromosome and chromosome after replication of genetic material. You see, genetic material has to double before cell division occurs. Chromosome after replication consists of two sister chromatids, the identical copies formed by DNA replication of a chromosome. A sister chromatid may also be said to be one half of the duplicated chromosome. A pair of sister chromatids is called a dyad. Each chromatid consists of DNA and associated with it proteins. Also, chromosome has centromere at its waist. The centromere appears as a constricted region of chromosomes and plays a key role in helping the cell divide up its DNA during cell division, mitosis and meiosis. I will cover these cellular processes in the next lecture, number 11. A typical human somatic cell contains 46 chromosomes. And each of human sex cells, gametes, contain 23 chromosomes. Thus, when two sex cells meet, sperm cell and egg cell, they form a new organism in which each somatic cell will contain 46 chromosomes. You got 23 chromosomes from your mother and 23 from your father. Most animals have between 10 to 50 chromosomes. Some crabs and ferns have up to 1,000 chromosomes. Karyotype, a normal chromosomal constitution of an organism. In different species, the amount of chromosomes varies. However, keep in mind, most bacteria have a single chromosome, regardless of their species. Here are karyotypes of different species. Notice the dogs and wolves that belong to the same species but different subspecies. Therefore, their chromosomal constitution or karyotype is the same, 78. Cow and goats are different species but have the same number of chromosomes in their karyotype. Thus, karyotypes do not show how species related to each other. Just look at the human and sable antelope. Same number of chromosomes, but this does not mean we are close relatives. Or look here. Human karyotype is 46, and potato karyotype is 48. Close numbers, but this does not mean we are close relatives with potato. Same things with rice and snails. Normally, all your cells, except eggs and sperm cells, have a set of the same 46 chromosomes. The chromosomes are grouped into 23 pairs. Each pair has 2 chromosomes, one coming from each parent. You may develop certain genetic disorders if your cells are missing whole chromosomes or pieces of chromosomes, or have extra chromosomes or extra pieces of chromosomes. When we examine chromosomes, we differentiate between autosomes and sex chromosomes. Autosomes are any chromosomes that is not sex-determining chromosomes. You can see 22 pairs of them on this slide. This is a karyotype of a human. Sex chromosomes are chromosomes which determine the sex of an individual organism, either it is a male or a female. You can see one pair of these chromosomes. Each is designated by a letter X and Y. If you are born with XX chromosomes, biologically you are considered to be a female. If you are born with XY chromosomes, biologically you are considered to be a male. Look close at this human karyotype. Is there anything unusual about this? Not really. There are 22 pairs of autosomes and 1 pair of sex chromosome. This makes it to be 46 chromosomes. Is this a karyotype of biological man or biological female? It is a karyotype of a biological male, as you can see from the sex chromosome composition. Now take a look at karyotype of this individual. Do you see anything unusual here? Yes, this is not a typical human karyotype. Look at a pair 21. There is an extra chromosome. Having too many or too few chromosomes can cause serious problems with health, growth, and normal development, such as Down syndrome. Extra chromosome 21. It is a human karyotype with a chromosomal mutation. This is a Down syndrome karyotype. I covered chromosomal mutations in the lecture number 14. The nucleolus is a spherical structure found in the cell's nucleus, whose primary function is to produce and assemble the cell's ribosomes. Look at the small black circles on the diagram in front of you. This is ribosomes. It may sound strange, but those little grains can read. Ribosomes are super important in the cell because they are the ones that produce proteins in the cell. It happens because they are capable reading instructions that come from DNA molecules via messenger RNA. We will cover this in a couple of lectures later on. As you remember from lecture number 9 titled Biopolymers, without proteins there will be no life. Both prokaryotic and eukaryotic cells contain thousands of tiny ribosomes, which are made of RNA and protein. When viewed by a very high magnification, ribosomes show up as a fine spherical specks dispersed throughout the cytoplasm. In eukaryotic cells, some of them attach to the endoplasmic reticulum of the cell. In prokaryotic cell, some of them attach to the cell membrane, since prokaryotic cells do not have endoplasmic reticulum, of course. Those ribosomes that attach to endoplasmic reticulum or plasma membrane, known as membrane-bound ribosomes. And those that float in the cytoplasm of the cell, known as free ribosomes. But all of them preoccupied with one of the most important processes, protein synthesis. Ribosomes are protein factories. Chemically, a ribosome is a combination of spherical type of RNA, unsurprisingly called ribosomal RNA or rRNA, and protein. One method of characterizing ribosome is by S, or Swertberg's units, which rate the molecular size of various cell parts that have been spun down and separated by molecular weight and shape in a centrifuge. Heavier, more compact structures sediment faster and are assigned a higher S rating. Combining these methods of analysis with high-resolution electron micrography has revealed that the eukaryotic ribosome, which has an overall rating of 80S, is actually composed of two smaller subunits, 60 and 40S. They fit together to form a miniature platform upon which protein synthesis is performed. The prokaryotic ribosome, which has an overall rating of 70S, composed of two smaller subunits of 50 and 30S. Sometimes students ask why 50S and 30S is make-up structure that is 70S, not 80S. This is because they miss the earliest point I tried to make. This is not an arithmetical calculation. This is centrifugation process. When you spin down two subunits attached together, they fall down to the bottom of the test tube with the rate of 70s. When the subunits detach from each other, the small subunits fall down with the rate of 30s, and the larger subunit falls down with the rate of 50s. Look what happened when we spin ribosome in a centrifuge. Pay attention to the rate of sedimentation. By the way, we are spinning now eukaryotic ribosome and eukaryotic ribosomal subunits. Of course, I overly simplified this process of sedimentation, but I hope this gave you an idea how we're able to assign the numbers that we just talked about. You might ask why we have to remember these numbers that are assigned to the ribosomal subunits. Well, these numbers are very important characteristics of the ribosomes, and this has very important practical implication for us humans. Here are the more realistic subunits of bacterial ribosomes, or we call them prokaryotic ribosomes, which are 70s ribosomes. I have no doubt you heard of antibiotics. If you watch my previous lectures, you know that antibiotics fight bacterial infections, not fungal or viral infections, but bacterial infections. How does this happen? The antibiotics bind to ribosomal subunits. When this happens, ribosomes of bacteria cannot produce proteins anymore. If bacterial cell does not produce proteins, it is unable to live anymore. It dies. The interesting thing is that the antibiotics are specific. For example, erythromycin, chloramphenicol, and tetracycline can bind only to 50s subunit, not 40s or 60s subunit with what our human ribosomes are made, but 50s, a large subunit of bacteria. Thus, these antibiotics are harmless to our cells while deadly to bacterial cells. Streptomycin and gentamicin binds to 30s subunit of bacterial cell, and this event kills the bacteria, while our ribosomes and cells are not harmed. Human pancreatic cell has a few million ribosomes. Why? Well, because our pancreas constantly has to produce a huge amount of protein molecules, important in digestion. Pancreatic enzymes, which are protein, has to break down sugars, fats, and starches. On top of this, our pancreas also helps our digestive system by making hormones. They are chemical messengers that travel through your blood. Insulin and glucagon are peptide hormones derived from proteins. Also, keep in mind that nuclei and ribosomes are not enclosed in a membrane. These organelles do not have membranous structures. This makes them different from other organelles we're about to cover. Yes, now time to cover organelles that belong to the cell and the membrane system. Those organelles that are surrounded by a membrane. As you will learn, not all organelles that are surrounded by a membrane belong to the cell and the membrane system. We already talked about nuclear membrane that surrounds the nucleus and forms nuclear envelope around the genetic information of the cell. Therefore, let's take a look at the endoplasmic reticulum. Endoplasmic reticulum, often abbreviated as an ER. ER is a network of membranous tubules and sacs called cisternae continuous with nuclear envelope. There are two types of ER, rough ER and smooth ER. Rough ER receives its name from the appearance of dense granules that tightly adhere to one side of the membrane, making it look a little like a coarse sandpaper. The rough ER consists of parallel rows of membranes surrounding deep channels. The small round bodies or grains are ribosomes. It is safe to conclude that a cell containing rough ER is highly involved in protein synthesis and probably secretes proteins out of the cell as fast as they found. It is involved in manufacture of cellular membranes and formation of sac-like lysosomes, vacuoles, and acrosomes. Acrosomes are structures in sperm cells. Smooth ER primarily found in cells that synthesize, secrete, or store carbohydrates, steroid hormones, lipids, or other non-protein products. Skeletal and cardiac muscles contain extensive network of smooth ER that store calcium and release it to trigger muscle contraction. Calcium is the final go signal for muscle contraction. One of the most important and main functions of endoplasmic reticulum is detoxification, which is removal of toxic material such as metabolic waste or drugs. Detoxification process is all done by smooth endoplasmic reticulum presented in the liver cells. Liver is an organ that is pretty much known for its detoxification and metabolizing drugs. It contains enzymes that catalyze a number of reactions that can make lipid-soluble drugs and metabolic waste return to the water. So these drugs and cell waste can easily be expelled out of the body. So as you can see, the smooth endoplasmic reticulum, not just a highway for biological molecules as it presented in many high school textbooks. This organelle has much more important functions within the cell. However, there is a busy traffic of molecules is moving out of the endoplasmic reticulum. Produced new molecules are constantly leaving the ER. They get packed in the vesicles which bud off from the endoplasmic reticulum surface and move on to the post office of the cell called Golgi apparatus, or some textbooks call it Golgi complex. Golgi apparatus consists of flattened sacs or cisterna that are not physically connected. This organelle that is on my diagram looks like a stack of pancakes, stores secretions. It modifies, refines, and ships proteins. Textbooks differentiate cis and trans phase in the traffic of balloon-like vesicles. Cis phase is near the endoplasmic reticulum, arrival lumen. The vesicle upon arrival fuses with the surface of Golgi membrane, releasing its content. Trans phase gives rise to the vesicle departure. Membrane appear to balloon out, forming vesicles that then pinch off to become closed sacs. From trans phase, vesicles travel to the place where they need it, for example, building up cell wall membrane. This is why Golgi apparatus often referred to as post office of the cell. So far, we looked at the relatively large structures of the cell. Now I want to bring your attention to the small sacs that are roughly spherical. They play an important role in cleaning up the inner of the cell. These structures named lysosomes. On this diagram, they are shaded yellow. Lysosomes are organelles that are found in animal cells that are responsible for digestion and waste disposal. Lysosomes contain enzymes that break down large molecules and destroy worn-out cell structures. Lysosomes are bags of powerful hydrolytic enzymes packed and synthesized by Golgi apparatus. These enzymes become activated at pH 5. So lysosomes have a hydrogen pump that pumps hydrogen ions into the organelle to reach the desired pH. If the digestive enzymes were floating free in the cell cytoplasm, the cell itself would be quickly digested. Christian de Duve, who first described lysosomes, called them suicide bags. His poetic fancy was not entirely unwarranted, since lysosomes sometimes actually serve the function of destroying the cells that bear them. If the enzymes of the single lysosome is released into a cell, there will be little change in the cytosol, but a massive enzymatic discharge by many lysosomes can be fatal to the cell. When this happens, this is not necessarily a treacherous act. Sometimes the destruction of cells is a normal part of embryonic development or is otherwise beneficial to the organism. For instance, lysosomes might destroy superfluous cells that was not functioning well, or one in the part of the body that is undergoing reduction as part of the developmental process, such as tissue between fingers in a developing hand. Then the lysosome open up and do their thing, and white blood cells clean up what's left. Through the coordinated release of lysosomal enzymes, number of important developmental changes occurs in varied multicellular organisms. As a tadpole matures into a frog, for instance, it loses its tail due to the lysosomal destructions of the cells contained in the appendage. Lysosomes have also specific functions in a healthy active cells. After phagocytosis, say after the ingestion of an invading bacterium by one of the body's own white blood cells, a lysosome will fuse with the digestive vacuole and empty its content into the vacuole, aiding to the digestion or to whatever have been captured. Obviously, such dangerous chemicals must normally be kept from spilling into the cell's interior, so the membranes of lysosomes are very tough. In fact, the toughness led to the discovery of the organelles. Certain organelles, researchers found, resisted all but the most vigorous attempts to rupture them. They could always be found intact after the cell was ravaged by a blender. When the mysterious particles were investigated with the electron microscope, they found them to be composed of little bag-like bodies. They were, in fact, lysosomes. Later, lysosomes were found in nearly all types of animal cells. Interestingly, there is some disagreement about whether they exist in plant cells. To a certain extent, it appears that different groups of people look at plant cells and animal cells. So similar organelles are sometimes known by different names. It is possible that some of the plastids found in plant cells are not much different from the lysosomes found in animal cells. In recent years, evidence has been accumulated that points to a role of lysosomes in the regular turnover of cell components. It seems that lysosomes destroy various cell organelles at fairly constant rate. Presumably that those parts can be replaced and cells can remain forever young and healthy. For example, in some tissue, mitochondria are replaced about every 10 days. Electron micrographs have occasionally shown a mitochondrion or half digested remains of a mitochondrion enclosed in what seems to be lysosome. Meanwhile, new mitochondria are produced by the division of old mitochondria. Lysosomes might thus be important in process of aging, a process that may not concern you now as much as it will later on. Finally, there are a number of genetic disorders in which some essential lysosomal enzymes are altered or absent. In Hurler syndrome, there is a lack of enzymes that usually breaks mucopolysaccharides. As a result, bone cells contain huge lysosomes swollen with mucus-like substance. This causes bone deformities. In Tay-Sachs disease, there is a lack of lysosomal enzymes that normally breaks down lipids in cells surrounding nerve cells. This causes nervous system gradually drown in lipids, and this affects motor skills, causes loss of vision, paralysis. Toddlers die before 4 years old. In Pompe disease, there is a lack of enzymes that normally breaks down glycogen. Glycogen builds up in cardiac muscle and liver cells, eventually causing heart failure. The term vacuole is rather general. It refers merely to any membrane-bound body with little or no inner structure. Vacuoles generally hold something, but their contents are very widely depending on a cell and an organism. Plant cells generally have more and larger vacuoles than do animal cells. In many types of plant cells, the vacuole dominates the central part of the cell, crowding all other elements against the cell wall. White adipose tissue cells have large vacuoles and low number of mitochondria. They have the ability and space to store lipids in the form of triglyceride droplets. This stops this fatty acid from circulating in the blood, which can cause inflammation of the blood vessels and arterial plaque buildup. Plants' vacuoles are filled with fluid, the cell sap, which is primarily water, with different substances in solution or suspension. The solutes may include the atmospheric gases, inorganic salts, organic acids, sugars, pigments, etc. Sometimes the vacuole is filled with a water-soluble blue or purple pigments, anthocyanins, which give color to blue or purple flowers. The large watery vacuoles of plants serve another curious purpose. While all animals have excretory system for getting rid of cellular and metabolic waste, plants lack such systems. Instead, metabolic waste and other poisons are sequestered in the central vacuoles, frequently forming crystals. Such plants store a large amount of waste poisons in their vacuoles, and it has been suggested that this serves a purpose device against herbivorous. Of course, if herbivore should eat the plant and crush the cell, it will not only get food, but stored poisons. Let's not forget another important function of vacuoles in plants, that is physical support. High concentration of dissolved substances in the vacuole can cause water to rush into the cell through the process of osmosis. This increased fluid pressure inside the vacuole can cause the cell to enlarge a bit and push out the cell wall. This process is responsible for the pressure that allows stems, flowers, and other parts of the plants to stand upright is due to primary turgor pressure. Wilting is a result of a loss of turgor pressure. And this is why some grocery stores keep some lettuce and vegetables under misters so the water will keep them turgid. In animals and humans, adipose tissue cells have large vacuoles and low number of mitochondria. They have the ability and the space to store lipids in the form of triglyceride droplets. This stops the fatty acids from circulating in the blood, which can cause inflammation of the blood vessels and arterial plaque buildup. Let's summarize now the functions of the vacuoles. In both animal and plant cells, vacuoles' function is nutrient storage. This is function number 1. In plants, it is waste management. The vacuole retains waste products and degrades them with digestive enzymes, just like the lysosomes in animal cells. The third function in plants is herbivore deterrence. Poisons that accumulate inside the vacuoles of some plants make a powerful deterrent to herbivores. Also, we can say that vacuoles indirectly play a role in sexual reproduction, since the vacuoles may contain a variety of pigments that give some flowers their red, blue, purple, or other colors. This attracts birds and insects that help the plant to reproduce by transferring pollen. And the last function is physical support in plants. On this, I will stop talking about the organelles that belong to the endomembrane system of the cell, and I will start covering autonomous cell organelles. Autonomous means independent, so I will cover those organelles that can function independently from endomembrane system of the cell. Mitochondria, peroxisomes, and in plants it is chloroplast. Also, I will cover centrioles, endoskeleton, cilia, and flagella. Mitochondria are not a part of endomembrane system. Mitochondria are known as the powerhouse of the cell because mitochondria are sites of cellular respiration. It generates ATP molecules by extracting energy from sugars, fats, and other fuels with the help of oxygen. Mitochondria, singular mitochondrion, is a semi-autonomous organelle, meaning they do not depend on the endomembrane system for its replication and function within the cell. Here is the etymology of the word semi-autonomous. Semi means partially. Autonomous means independent or self-governed. Why? Or how come mitochondria are independent organelles? To understand this, we better take a look at the anatomy of a mitochondrion. And the best thing to understand the anatomy of something is to make a diagram of it. So let's do this together. First, we draw outer membrane of mitochondria. Here it is. Let's not forget to label it. We always have to label diagrams. Now we want to draw inner membrane of the mitochondrion. This should not be hard to do. The inner membrane is wrinkled. Here it is. We also will label it. Inner membrane. The inner membrane forms numerous folds called cristae, which extend into the interior of the organelle called matrix. To show the matrix, we can shade it with bluish color. We also should label this. Matrix. The matrix of a mitochondrion is a mitochondrial internal space enclosed by the inner membrane. It contains a large variety of enzymes that involved in production of the ATP molecules, most important energy molecules of a cell. Our mitochondria allows us to convert energy contained in the chemical bonds of food we eat into ATP. We will talk about this molecule in a bit from now, but now we will draw the most interesting part of the mitochondrion that might surprise you, the mitochondrial DNA. Yes, mitochondria contains its own DNA molecules. It means this organelle capable to reproduce on its own independently from endomembrane system of the cell we covered earlier. Mitochondrial DNA is small and circular. Here it is. In humans, mitochondrial DNA contains 37 genes, all of which are essential for normal mitochondrial function. And here is more surprises for you. Mitochondria contains its own ribosomes. Their membrane proteins are made not by the endoplasmic reticulum, but by free ribosomes. By the way, what ribosomes you think mitochondrion contains? 80S or 70S? It is 70S ribosomes, like in bacterial cells. Each mitochondrion has its own free ribosomes and mitochondrial DNA for its growth, replication, repair, etc. Mitochondria are powerhouses of animal, plant, and fungal cells. Almost all eukaryotic cells have mitochondria. Mitochondria are very important organelles of the eukaryotic cells because it produces super important macromolecules called adenosine triphosphate, ATP. These molecules are advantageous for the living cell. The more ATP it has, the more things cell can do. The most of the processes within the cell pays with energy stored in the ATP molecules. In each of these molecules, the adenosine is attached to 3 phosphate groups. The last phosphate group is attached to the molecule by high energy bond. When the bond is broken and energy is released, this energy is used by the cell to conduct numerous tasks, moving various materials around, fuel chemical reactions, turning on some processes, etc. So every time a cell needs energy, it gets it from the ATP molecules. When phosphate is taken from ATP molecule, it becomes adenosine diphosphate. As you remember, di means 2. Sometimes when there is not enough ATP molecules, the cell breaks the bond in the ADP molecule. So the adenosine diphosphate becomes adenosine monophosphate. As you remember, mono means 1. The second bond has less energy than the high energy bond, but enough to do the job. When a cell becomes desperate for energy, even the AMP could be used, leaving adenosine as a single molecule. A question: when AMP is broken into adenosine, does energy get released? Yes, since covalent bonds contain energy. However, there is much less energy released than when the other 2 bonds get broken. What cells do you think need to have many mitochondria? Cells with high energy needs have many times more mitochondria than cells with low energy needs. The muscle cells use many of ATP molecules for its contraction. Therefore, they require many mitochondria to be present. However, liver cells require twice as much mitochondria than muscle cells because these cells are highly metabolically active and regenerative. ATP molecules is like money for the cells. The more ATP cell has, the more things it can do. Think of a mature red blood cell. This cells is just a bag with hemoglobin molecules that bind to oxygen and release it to the body tissue. Red blood cells just flow in the stream of plasma. It does not need any ATP energy, therefore it does not have them. White adipose cells is functioning in the body as a storage for the energy. It holds fat molecules. For its metabolism, it needs about 100 mitochondria per cell. In contrast with muscle cells that are filled with mitochondria, it needs a lot of energy to perform the job. And highly regenerative liver cells need twice as much ATP energy as muscle cells. Because mitochondria do not depend on the endomembrane system of the eukaryotic cell, it has been thought that during evolution, these organelles had been living as an independent organisms until the large cells engulfed them. But for some reason did not digest them. And this is how mitochondrion found its home and the large cell acquired its tenant, which started to pay rent with ATP molecules. This type of relationship is common in nature and known as symbiosis, a close association between two different types of organisms, in this case, mutually beneficial relationship. In the scientific literature, this view on mitochondria is known as endosymbiotic theory. Endo means inside, as you might remember. The process of making ATP molecules is known as cellular respiration. In the presence of oxygen, eukaryotic cell makes 36 molecules of ATP. For this process to happen, eukaryotic cell requires oxygen molecule, sugar, usually glucose, investment of 2 ATP molecules, inorganic phosphate, and specific enzymes. The process is known as aerobic respiration, respiration using oxygen. Most of this process happens in mitochondria. Without the molecule of oxygen, only 2 molecules of ATP could be made. The process is known as anaerobic respiration. All of us are familiar with the pain the next day after excessive exercise. It occurs if we have a physically inactive lifestyle before exercise. Consequently, we complain of sore muscles. The pain occurs because cells are running out of available oxygen, and a switch occurs between aerobic respiration to the type of anaerobic respiration known as lactic acid fermentation, in which only 2, not 36 ATPs are made per each glucose molecule. It is better than nothing. However, during this process, lactic acid is formed as a byproduct. Its molecules accumulate in the muscle and cause pain. I hope now you will be able to answer the question why we, animals, plants, fungi, and even most of bacteria do need oxygen. So what is an answer? We need oxygen to make ATP molecules upon which we rely for our cellular processes. In the absence of oxygen, our mitochondria will not be able to produce ATPs. So what will happen in case we do not have access to oxygen? Body cells will quickly run out of ATPs, and the major biochemical processes in our body will cease, which in turn will lead to the cellular death and to the death of a whole organism. In fact, we can survive in the absence of the oxygen only for 3 minutes. Some animals are capable of slowing down the rate of their metabolism and can survive without access to oxygen much longer than we would. Even though the bacterial cells have very different cellular organization, their major biochemical reactions are similar to ours, so their cells need oxygen, especially if the bacterial cells are metabolically active. Now I want to clarify a few things about ATP molecule, since one might get misconception concerning these molecules. In all textbooks, mitochondria are often referred as a powerhouse of the cell because it produces ATP molecules. There is this is not very wrong, but one might think that ATP has a lot of energy. Do not get confused. The third bond has more energy than the second and first bond, but it does not mean there is a lot of energy making the bond strong. This is not a strong bond. In fact, it's about to break at any chance it gets. Look at the molecular structure of the ATP molecule. You see adenine, sugar called ribose in its molecule. And here, the 3 phosphate groups. Now look at the charges each phosphate group has. The charges are negative and they're crowded together. Earlier in the course, you learned that the alike charges repel. Their repulsion contributes to the instability of this region of ATP molecule. The triphosphate tail of ATP molecule is a chemical equivalent of a loaded spring. When ATP hydrolyzed in the test tubes, the release of energy merely hit the surrounding water. In the cells, that will be an inefficient and dangerous use of valuable energy resource. With the help of the specific enzymes, the cell is able to couple the energy of ATP hydrolysis directly to metabolic process that needs it. Nearly all cellular work depends on ATP that energizes the molecules by transferring phosphate groups. For example, during muscle contraction, ATP molecules transfer phosphate groups to contractile proteins. Mitochondrial DNA in humans is always inherited from a person's mother, not father. Thus, we get more genetic information from our mothers. To understand why, we should take a look at the anatomy of a sperm cell. Sperm cell contains 50 to 75 pieces of mitochondria in its midpiece. The structure and function of the sperm mitochondria are essentially similar to mitochondria in body cells. The sperm mitochondria produce energy for the movement of the sperm. After all, sperm cells have to swim all the way upstream. Thanks to the mitochondria, the sperm cells known as the best swimmers. Sperm cells use flagella to propel through the environment. Flagella beating drives sperms through the female reproductive tract and is vital for reproduction. There are no mitochondria in the head of the sperm cell. The head contains nucleus with its DNA molecule. A structure called acrosome attached to the head of the cell. Acrosome carries the enzyme capable to dissolve egg coat, but one sperm does not have enough enzyme to do the work. This is why having a low sperm count can make it more difficult to conceive naturally, although successful pregnancies can still occur. In order to get fertilized, the egg requires many sperm cells to break the egg coat, so one lucky sperm can inject it with its DNA molecule. However, the meat piece is not entering the egg, so all father's mitochondria get destroyed. Thus, all of us inherit only mother's mitochondrial DNA. The shape of the muscle might be inherited from the father, but its strength is inherited from the mother. You might have heard of trichomoniasis or trich. It is a very common sexually transmitted infection. In women, trichomoniasis can cause foul-smelling vaginal discharge, genital itching, and painful urination. Men who have trichomoniasis typically have no symptoms. Risk factors include multiple sex partners and not using condoms during sex. The causative agent is not bacteria, not virus or fungi, but a protozoan parasite. Scientific name for the species is Trichomonas vaginalis. It is an eukaryotic organism that does not have mitochondria. Instead, it has organelles called hydrogenosomes. Hydrogenosomes are membrane-enclosed organelles found in some anaerobic ciliates, flagellates, and fungi. Hydrogenosomes are highly variable organelles that have presumably evolved from protomitochondria to produce ATP in anaerobic conditions. As name implies, hydrogenosomes utilize molecular hydrogen instead of molecular oxygen in the synthesis of ATP. However, this is not typical for eukaryotic cells. This is an exception from the rule. Another cell organelles that are membrane-bound but do not belong to cells and the membrane system are peroxisomes. On the diagram in front of you, you can see them as small red circles. In reality, of course, they do not have any color. Peroxisomes do not have its own DNA, but they are capable of replicating themselves by splitting into two. Peroxisomes are found in great variety of organisms. They are found in plants, animals, and protists. In animals, they are most common in liver and kidney cells. Peroxisomes appear very dense bodies with unique crystalline score of tiny tubes, which cause the organelles to stand out unmistakably when it is shown up in the electron micrographs. Peroxisomes resemble lysosomes, but contain different enzymes and are not produced by Golgi complex. They reproduce by enlarging and then splitting in two. Peroxisomes produce hydrogen peroxide in the course of detoxifying alcohol and killing bacteria. The principal enzyme of the liver and kidney peroxisomes is catalase, which helps break down hydrogen peroxide into water and oxygen. You can see this on the chemical equation on this diagram. The hydrogen peroxide is oxidized to water and oxygen, rendering the peroxide harmless to the cell. All the peroxisomes decompose fatty acids into two carbon acetyl groups, which the mitochondria then use as an energy source for ATP production. The phospholipids of the plasma membrane spread out over the cell skeleton like butter on the slice of bread. Function of cytoskeleton include structural support. It is an obvious function. Cell motility. Do not confuse this word with mobility. Mobility is all about portability. Oh, I think so. Maybe I am wrong. Motility means self-induced movement and cell regulation. There are many processes that cell has to regulate inside and outside of its unit. Cytoskeleton is composed of microfilaments, intermediate filaments, and microtubules. The last on this list is the largest ones. Cilia and flagella are fine hair-like movable organelles found on the surface of some cells. Actually, although cilia and flagella appear to be outside the cell, they really are not. The cell membrane protrudes to cover each cilium. Thus, we can consider them to be outpocketing of the cell proper. Cilia and flagella are structurally almost identical. Nearly every human cell has a single non-motile primary cilium. Its function in some cases still a mystery, but apparently many of them are sensory, serving as antennas, like a cell's antennas for motility nearby conditions. In the inner ear, they may play a role in the sense of balance. In the retina of the eye, they are highly elaborate and the light-absorbing part of the receptor cells. And in the kidney, they are thought to monitor the flow of fluid as it is processed into urine. Sensory cells in the nose have multiple non-motile cilia that binds other molecules. Motile cilia are less widespread, occurring in the respiratory tract, fallopian tubes, internal cavities of the brain, and short ducts associated with testes. There are maybe 50 to 200 cilia on surface of one cell. They bear in waves, sweep across the surface of an epithelium, always in the same direction, propelling such materials as mucus, an egg cell, or cerebrospinal fluid. After cilia completes its power stroke, it is pulled limply back by a recovery stroke that restores it to the upright position, ready to flex again. Flagella beats differently. A flagellum has an undulating motion that generates force in the same direction as flagellum's axis. A cilium and flagellum has a core of microtubules sheathed in an extension of the plasma membrane. 9 doublets of microtubules, the membrane of each pair shared part of their walls, are arranged in a ring. In the center of the ring are two single microtubules. This arrangement, referred to as the 9+2 patterns, is found in nearly all eukaryotic flagella and cilia. The outer microtubule doublets and the two central microtubules are held together by flexible wheels of cross-linking proteins, the structure called radial spoke. As you see, each outer doublet also has pairs of side arms spaced along its length and reaching towards the neighboring doublet. These are motor molecules. They are called dynein arms. These dynein arms are responsible for the bending movement of cilium and flagella. The ATP provides the energy for these movements. The microtubule assembly of cilium and flagellum is anchored in the cell by a basal body, which is structurally identical to centriole. Centrioles are short cylindrical bodies, each composed of a circle of 9 triplets of microtubules. They form mitotic spindle during cell division. We will talk about mitotic spindle in our next lecture on cell division. As you just learned, unpaired centrioles form basal bodies of cilia and flagella, and they control the direction of cilia and flagella movement. Centrioles replicate autonomously like mitochondria and peroxisomes. Like cilia and flagella, centrioles also made of microtubules. The difference is that they contain 9 sets of triplets and no doublet in the center. Here is another organelle in animal cell, not in plant cell. This organelle called centrosome. Centrosome of animal cell are pair centrioles. 9 sets of triplets microtubules arranged in a ring. This function of centrosome in an animal cell is to regulate cell motility, adhesion, and polarity in interface. Centrosomes facilitate the organization of the spindle poles during mitosis. Perhaps these functions of centrosomes do not make much sense to you now, but believe me, in the next lecture on cell division, the lecture number 11, you will understand what this means. Now we will have to move with the lecture and see the difference between animal and plant cell. The distinction between plant and animal cell based on the organelles and structure they contain. Plant cells have a large central vacuole called a tonoplast. This large vacuole produces internal pressure against the cell wall, as I once mentioned. Plant cells also contain green pigment chlorophyll packed in an organelle called chloroplast. Chlorophyll traps the energy in light to be used in all important functions of photosynthesis. Chlorophyll absorbs red and blue light, reflecting the green segment of the light spectrum. Inside their double membranes, the chloroplast consists primarily of layers of flattened membranous discs, known as thylakoids. Each stack of discs is known as granum. Many grana are distributed through the chloroplast in orderly rows. Each granum is connected to its neighbors by membranous extension of the thylakoids. The surrounding matrix is called the stroma. The process of photosynthesis depends on the very precise structure of the components of the chloroplast. If chloroplasts are broken open, photosynthesis ceases abruptly. The rigidity of the plant cells is due to the non-living cell wall found bordering the outside of the less rigid cell membrane. The cell wall provides protection and support and is made up of 2 or more layers. The cell wall is composed mainly of cellulose and is more rigid. Animal cells do not have tonoplasts, chloroplasts, or cell walls. In the next lecture, you will learn how cells reproduce to form genetically equivalent daughter cells. As Rudolf Virchow put in 1855, where a cell exists, there must have been a pre-existing cell. Just as the animal arises only from an animal, the plant only from a plant.